Good morning. Good morning from Asia Society Hong Kong Center. I am Executive Director Alice Mong, and it's my pleasure to welcome all of you this morning and good evening in Texas. It is a delight for Asia Society Hong Kong to be collaborating with our center in Texas for this program this morning. Before I get started with the intro of our speakers and our moderator, just a bit of context from for today, May 29th, 2020. I think, unfortunately, we've reached over some sad milestones. I believe 100,000 in the United States have passed away due to COVID, and over 5 million worldwide cases. We are very fortunate in Hong Kong. Our case is still quite low, at 1,069, I believe, and with 4 deaths. And I know the Texas case is also quite low comparative to the other states in the United States, was over about 59,000 cases. So today we are really delighted that this collaboration with our Texas Center, partnership with our Asia Society Texas Center on this webcast. This is our Asia Society Hong Kong's coronavirus update, episode 15, and we started it in February, Valentine's Day, February 14th. With a professor from Hong Kong University, Professor Cowling. This is week 9 of the Texas Center webcast series on COVID-19, which explores various themes and topics impacting the larger community. Today, we have Professor Hongling Chen, is the founding member of the State Key Laboratory for Emerging Infectious Diseases at the University of Hong Kong. And the focus of his research is on RNA viruses, including emerging influenza and coronavirus. He has also established several research programs which focus on studying molecular determinants for cross-species of avian influenza viruses and coronavirus. And in Houston, in Texas, we have Dr. John Cook, who is the chair of the Department of Cardiovascular Sciences, the director of the Center for Cardiovascular Regeneration, and director of the RNA Therapeutics Program at the Houston Methodist Research Institute. The focus of his research is on regenerative medicine, and during the COVID crisis, he has turned his group toward generating an RNA vaccine against SARS-CoV-2. And our moderator today is Eddie Allen. He is co-founder and senior partner of Eagle Global Advisors, an investment advisory firm based in Houston, Texas. Eddie serves as our chairman of the Asia Society Texas Center and is on the board of the Asia Society Global Organization. Following the conversation, we will have some questions from the viewer. So please share your question through the discussion area via Facebook and YouTube comment boxes. So please, thank you, and I'm going to turn it over to our able moderator. Thank you.
Thank you, Alice. It's so nice to see you virtually across the world. Amazing, this technology that we have today. And hopefully this technology is going to be able to help us defeat this virus. I'm very excited to hear from Dr. Chen and Dr. Cook, perspectives from different sides of the world on what's some of the therapies that are being and vaccines that are being developed for this virus. I thought I'd start with Dr. Chen. Please tell us about the state lab for emerging infectious diseases that you are part of in Hong Kong, and tell us about the vaccine efforts that your lab is involved with. I understand that your lab is one of 5 that the Ministry of Science in China has targeted or highlighted for its emerging technologies. What can you tell us about what's going on in your lab there?
Thank you for having me here today. So I'll start introducing the state laboratory for emerging infections. So it was established in 2005, and right after the outbreak of SARS, and Hong Kong University scientists have did a nice job during in a response to the SARS. So because of that, So they say, okay, probably necessary to establish a lab specifically focused for a rapid response to the emerging infections, like SARS or other potential emerging infections. So it's due, it's because of the background. So it's that laboratory. And since that, we have been working on SARS, avian flu, swine flu, and also we did a lot of work on coronavirus, which is mainly in Middle East, the MERS. And this time we also have response aggressively and swiftly to COVID-19. So in our lab, we have a long history of working on influenza. So for my work, we also, based on influenza, we develop a system, and this system is potentially can be used as a vaccine platform. So this time when it happens, the emergence of this coronavirus, we immediately make a vaccine C in about 2 weeks' time. And after that, we are collaborating with some manufacturer on mainland China, try to move it to a clinical trial, and hopefully it can be tested and eventually it can be used in humans. So that's what we are doing in the lab and the vaccine project. And after that, we have been in recently collaborating with CEPI. And also we are doing some laboratory work for animal trial in the UK public health.
And the virus that you have developed to combat COVID-19 is based on live attenuated viral strands. Is that right? So is this, can you describe the technology that you're using and its advantages or cons to treat the virus?
Yeah, so the system we are using is flu-based, means it is based on a flu virus. So we are working on flu, try to understand the pathogenesis of influenza virus. So we knock out one of the very important virulent factor from influenza virus genome, and they make the virus fully attenuate. But usually when you attenuate, it's very difficult to produce. But our scientists, they identify some mutations which enable the attenuated influenza virus can be reproduced in the vaccine producing system, like eggs and some cell lines. So this time we say, okay, we have knocked out one of the virulent gene. So there's a space, so we can put other antigen into that space. So we have tested that in using the MERS coronavirus, and it's very effectively in animal model. So this time we do the same thing. We put one of the antigen called receptor binding domain from COVID-19 into that place. And because it is in the influenza virus system, so we can produce it in egg or in cell lines.
This is a traditional vaccine production technology, correct? So it's well known. And
yeah,
yes.
Flu vaccine.
Right.
So if you have a facility can produce flu vaccine, it can be used to produce this vaccine for COVID-19.
So it could potentially be used around the world, since these sorts of production facilities are available around the world, right?
Yes. So that's now we had a meeting with Gates Foundation. They say, okay, some countries they don't have their vaccine program, but they have flu vaccine facility. They are hoping if they can use our vaccine to produce, just fine.
Yeah. How soon will you be able to test your virus in humans?
So we have
your vaccine.
We have went through some preclinical studies. So now it's in the process to get the regulatory approval from mainland China. And we are targeting, if everything goes smoothly, we may be able to start the phase 1 clinical trial in July or around that time.
Well, that would be fantastic. And if everything goes well, when do you think your vaccine could be available for production?
Once the clinical trial phase 1 and phase 2 finish, And yeah, that will be some difficulty to do phase 3 in China because there are basically no many cases right now. But once there, and we hope if we can do by other way or to collaborating with other places to do the phase 3, if it's possible, we hope it can be ready by even not in huge amount of vaccines can be ready for this winter because we are worrying virus may come back and another wave during the winter time.
That would be fantastic. Well, let me turn to Dr. Cook in Houston to kind of fill us in perhaps on the efforts that are going on at the Houston Methodist Hospital and the research environment here in Texas. And for the viewers that aren't as familiar with Houston, Houston has the largest medical center in actually the United States. And it's sort of not as well known as perhaps some of the other medical centers in America, but a lot of interesting research is going on here. And Dr. Cook, can you introduce us to Methodist and what you're doing there, also on the COVID vaccine?
Thanks, Eddie. I really do appreciate the opportunity to be on this call. And the first thing I'd like to say is this meeting that we're having tonight is really emblematic of the kind of cooperation and collaboration and communication that's occurring around the world between scientists from every country. It's been incredible, the amount of information that's coming out. Everyone is sharing their data, and we're all working together to try to beat this thing. And for me, it's been, as a physician, scientist, it's been really heartwarming to see the kind of openness and collaboration that is happening around the world. In the Texas Medical Center, you're right, Eddie, I think the Texas Medical Center might be the biggest medical center in the world. It certainly is. It's the biggest in the United States, and there is a lot of exciting work going on. With the pandemic, our research institute made the decision to stay open because we had a lot of COVID-19 related projects going on. Other universities closed down and there's good reasons to do that. But we were able to maintain our work by social distancing, by putting in place other measures to protect the staff, protect people, staggered work schedules, having people come in on the weekends, spreading people out, testing everyone at the checkpoint for the temperature, that kind of thing. So we were able to keep operations going, and as a result, we've actually made some good progress in a number of key areas. One of the COVID-19 related projects that has gone very well is our convalescent plasma project. This is a project where we take blood from patients that have recovered. From that blood, we isolate the plasma, which has antibodies that allowed that patient to recover from COVID-19. Well, those antibodies can be effective against that plasma containing the antibodies can be effective against the disease. So it can be infused into people that are very sick. We've now done this procedure in about 75 patients, and it looks very promising, certainly safe, and time will tell whether or not it is better than not doing so.
We're the first hospital in America that was got the approval to try that plasma treatment. Is that right?
We were the first hospital in the country to get that underway, and it's a great teamwork from our pathology department and our infectious disease group.
Now on the vaccine front, though, you're taking a very different approach than Dr. Chen's group in China, right? Can you tell us about that approach?
I think Dr. Chen's approach is outstanding. We have a different approach. Dr. Chen is using basically, if I understood you, Dr. Chen, a weakened form of the influenza virus and putting into it a piece of the coronavirus that will create an immune response. And then when the real virus comes, the person is prepared. They've seen part of that virus, and now they can mount an immune response. We're doing a different approach. We're using a very small portion of the virus. Actually, that's RNA, which is the biological software that encodes proteins of the virus. So we're not putting in the virus, we're not putting in actual proteins from the virus. We're actually putting in into the body RNA that is the biological software encodes the proteins. We let the human cells make a protein, the spike protein of the COVID-19 virus. And the idea is that if the cells start making that spike protein, it'll get recognized by the immune system as being foreign, and an immune response will be generated.
Now, this is potentially a radically, exciting technology, but it's never produced a commercially viable virus, from what I understand. But what are the pros of taking this approach to?
Well, just to respond to that, Eddie, it's a very new approach, and the reason it hasn't, we haven't developed a vaccine yet with RNA is because it is so new. The traditional vaccines have been as Dr. Chen is doing, some form of a weakened virus. His is genetically modified in a way that makes it even more interesting. But weakened viruses or killed viruses, those used to be the traditional kinds of vaccine, like the polio vaccine, the Salk vaccine. Now, increasingly, there's other approaches. People are making proteins that are from the virus, or they're making, in our case, we're making RNA or DNA vaccines, DNA that encodes a piece of the virus. So there's different approaches. We need different approaches. There's about 145 vaccines now that are under investigation, and on average, only about 1 out of 100 make it all the way through the rigorous trials that are necessary for FDA approval. So we need 145 candidates to get something useful. We need a few shots on goal.
Well, I know we have had a number of vaccine candidates get a lot of press here in the US. There's the vaccine from Moderna. There's a vaccine from Oxford University, a vaccine from Inovio. Do you feel that, one of these vaccines might be more promising than another, or is there too early to tell at this point?
I think it's too early to tell, although there are some promising data. The Moderna vaccine, which you mentioned, is an RNA vaccine. Our RNA vaccine is different. We think it has some potential in a different way than the Moderna RNA vaccine. The RNA vaccine from Moderna, though, does look promising in early clinical trials. There was an immune response that was generated by the RNA vaccine, and in fact, in some cases, the response was very strong, and individuals felt like they were sick, which is an indication that maybe the dose was too high. So I think in the next clinical trial, they'll probably back off on the dose, but it seems like it's working.
Well, that's very exciting. Let's perhaps go back to Dr. Chen. Dr. Chen, there are also many different vaccine efforts going on in China. Perhaps you could comment on what you think are some of the more promising candidates, and are there have they started human trials with any of the vaccines in China?
Okay, so as you probably heard from media, there are 5 strategies adopted by Ministry of Science and Technology, and one of them is the inactivate virus. So it's a traditional technology. You use just cultural virus, you kill the virus, and then use that to immunize humans. It's all technology is very mature. So I think there are 3 or 4 companies already did the phase 1 or even phase 1 plus phase 3 clinical trial. And another technology is using adenovirus 5, subtype 5. And it's similar, you insert one of the COVID-19 antigen into the adenovirus 5, subtype 5. And it's a attenuated virus, and then use that as a vaccine to immunize human. And there are also one mRNA vaccine, like Dr. Koo just described, but probably different form of different, slightly different approach. Also, one DNA vaccine, it basically is a DNA vector. You express the antigen from COVID-19 and make it into a vaccine vector for immunization. Another one is our flu-based vaccine. So the adenovirus vaccine already in the phase. They published the phase 1 result in Lancet a few days ago, and they are now in phase 2, and 3 or 4 inactivation already finished the phase 1. So I think they are planning the phase 2. We are in the process to get approval for the phase 1. Hopefully, it will approve soon, and we can start try in July or around that time. And mRNA and DNA vaccine, I have not heard anything about the progress. Hopefully, they will move on to the clinical trial.
Dr. Cook, did you have a comment?
Oh yeah, I just Eddie, you mentioned Inovio, and Inovio has a different kind of vaccine, a DNA vaccine. Now I think the RNA vaccines are safer because the RNA vaccine is not going to get into your nucleus of the cell. It's not going to incorporate into your genome. DNA vaccine probably won't either, but it's possible. So the DNA vaccine is a little I think is not as safe as the RNA vaccine. That's a bias. I may be wrong about that. But I do want to make a plug for VGXI, our local Houston company, as the manufacturer of Inovio's vaccine. So here in Houston, we have a company that's making vaccines that are in clinical trials right now. It's VGXI. That company happens to be our partner at Houston Methodist Research Institute. They're working with us on the RNA vaccine. So we got quite a good effort going on here in Houston for vaccine development.
Well, let me ask you, I've learned that some vaccines might just protect the recipient from getting the disease, but actually might not be good for stopping the spread of the disease. That people could get the vaccine, it would protect them, but it wouldn't be strong enough to perhaps keep them from being a spreader. Is that an issue with these vaccines that you're working with, Dr. Cook?
Well, you know, I'm going to let Dr. Chen answer that question because he's got more expertise in this area. But I will say that you're right. You want a vaccine that's going to neutralize the virus, but also get rid of the cells that are generating the virus. So you need two. You need the antibodies. You need neutralizing antibodies, and you also need T cells that will go and get rid of the cells that are infected. And so you're absolutely right, Eddie. You need both, and it remains to be seen if any of these vaccines do both of those things. It's still early days.
Right, Dr. Chen, what do you about that issue with the viruses?
We agree with the comment from Dr. Cook, and also he mentioned about why this time we need multiple technology for this vaccine because it's a new virus, and we don't know which one will be working and go through all the way to the human application. So I think we for any vaccine we hope to achieve is it can prevent the disease and also can block the transmission. But we don't know all our vaccine now in different stage of development will be achieve both to prevent the disease and also to block the transmission. So based on some of the limited information we have seen from the literature, seems they all have some efficacy to alleviate the disease progression, but maybe still we are, I think people are still looking for if it can completely block the transmission. And based on of the result from literature, seems it's still challenging. Based on I think it's from monkey data, and seems it cannot be able completely block the transmission. And that so I think we are I think every vaccine is still working on and see from the preclinical we can get the data to that, and then also can be confirmed from human trials.
Now, apparently this virus is already perhaps mutated once, and that there's perhaps a little bit different strain. And what is the fear that it might continue to mutate and potentially defeat the viruses that are being developed for the current strains? Is that a concern, Dr. Chen?
Yeah, we also noticed that, and there are literatures, there are mutations. So basically, you will see the original strain earlier time from mainland China or Asian countries. a little bit different to now prevalence in Europe and US. And in particular, there's one other mutation called is 614G. And that one, because it's concerned, is because in the spike protein, and it's predominantly in US right now. And but I think we have tested those strains carrying that mutation in animal model. It doesn't cause more severe disease. And we are now testing our vaccine if it also can prevent the infection, also the disease progression with the earlier time strain and also this most recent strains. My feeling is like we are using antigen is derived from the receptor binding domain, and that mutation is outside that domain. So we are not really particularly concerned about that mutation. But we don't know if that mutation is making the virus more transmissible, but it needs animal data to support that. So we don't know those mutations, and we will see there will be more mutations or polymorphisms emerge from the circulating strains.
Well, that's good. So, sorry, Dr. Cook.
Sorry, I was just going to say, I think you asked a great question, and that is, could mutations of the virus, and viruses are mutating all the time, could that defeat the vaccine that is being developed? And I think Dr. Chen's response is very accurate. And what he is saying is that what we design our vaccines against parts of the virus that it's difficult for the virus to mutate because if they mutate those pieces, they may not work. So, for example, Dr. Chen and I have something in common. Our teams are going after the spike protein of the coronavirus, and that's the protein that is absolutely critical to let the coronavirus get into our cells. So the coronavirus can't mutate the spike protein too much, or it won't actually be able to get into our cells. So, nevertheless, though, it's a really good question, Eddie. And one of the things that the advantages that RNA vaccines have is that we can quickly change them. We can quickly change the sequence. We're just putting biological software into the cells, and we can change the sequence of the software and get respond quickly to a mutation if the mutation is important.
Well, let me ask both of you. One concern is that in America we're calling it the warp speed plan to get these viruses approved. But in some quarters, there's concern that we might be moving too fast. Obviously, we want to get a vaccine as quick as we can, but at the same time, in America, there's a segment of the population that's very suspicious of vaccines for whatever reason. How confident are you that the process to vet these vaccines will result in a safe and effective vaccine that doesn't cut corners, and when it is ultimately administered to people, that it's at the very least it's safe? Maybe I'll start with you, Dr. Cook.
Well, in the U.S., I think the FDA is still very tough. They've I would say they're helping. They're trying to accelerate the process, the regulatory process, but vaccines still have to go through phase 1, phase 2, phase 3. So phase 1 is trials in normal volunteers. Phase 2 is typically in people with disease or sicker. And then phase 3 are the large trials that are necessary to show that something works for sure. So the early trials are just for safety. The later trials are for safety and efficacy. The final trials are really to show, and when I mean efficacy, I mean that this vaccine is going to work, that it's safe and that it's going to work. So the FDA is not backing down on those kind of rigorous hurdles. They are allowing some things to happen in parallel. For example, Moderna began its phase 1 clinical trials prior to completion of some of its preclinical trials. So it was still doing animal studies while it's doing the early human studies. So I think, yeah, things have been sped up a bit, but they're still going to be rigorous. We're still going to have to go over those hurdles. And it's a good thing too, because we want products that are safe and effective.
Well, and also let me ask you, the elderly are more susceptible to something like this, and the elderly take a lot of drugs, and we don't know what kind of drug interactions there might be. Will they test those drug interactions before they release a vaccine to the public?
Well, that is a great question, Eddie, and I think that we have to take advantage of bioinformatics and epidemiological studies to get as much information as we can about those kinds of interactions. But the short answer is to your question is yes. That's the reason why phase 3 trials have to be large, because the FDA wants to see when a drug gets out into the general population, is it's going to have adverse interactions with other drugs. So that's why the phase 2 studies with small groups of people, 50, 100, 200 people, that's not enough. We need trials with thousands of patients to really show those kinds of interactions, see if there's adverse interactions with other drugs.
Let me ask Dr. Chen, what are the hurdles similar to getting a drug approved in China for use? Do you also have the similar safeguards as the FDA imposes here in the US?
Yeah, I think so. Because for any vaccine, the most important issue is about the safety, because we are going to use vaccine in healthy individuals. Drug maybe sometimes for clinical trial using in sick patients, but for vaccine, basically you are using for individuals who are healthy and try to prevent infections. So safety must be the higher priorities. So we have to go through the safety and then to try to see if it's effective to prevent disease, and also it can be used for all age groups. So I think it's a timeline. So that's why it always takes many years sometimes for a vaccine to be from the R&D to the human application. But this time is unprecedented speed, because of the collaboration, because of the work from previous experience. So multiple vaccines already in different stages of clinical trial. So hopefully it will move on more effectively. And one of the challenges will be the phase 3, because without phase 3, you cannot put into the human application. It means for vaccinate a general population. So phase 3 may be one of the hurdles, because in some places you don't have enough patients or infections to test the efficacy. There are still going on in UK, but it's also, I think, from the news you see that the cases are going down in Europe. US is still plateau, so we don't know if you can do it now, maybe okay, but later on maybe also decline. And if you don't have that virus still circulating, and phase 3 may be difficult to do. So there are also discussions about can we do human challenge? Yeah, but it involves a lot of ethic issues.
Let me just make sure I understand. So you say normally the phase 3 trial, you just give it to the patients and let them mingle in the general population, but we're afraid that there's not enough virus in the general population now to have a strong test. So you're actually talking about potentially giving the virus to some volunteers and exposing those volunteers deliberately to the virus. Is that ever been done before?
In influenza, it has been done in some places, like in Europe, they are doing human challenge for influenza. But influenza is a proven pathogen, and we know the pathogenesis. And for influenza, we have drugs. So it means if unfortunately a patient challenged with influenza and get into more severe disease, we have drug to treat.
But the problem is the COVID-19 is too lethal that to actually expose somebody would to expose them to death.
Very bad, but we don't have a drug, so that's the issue. But by still discussing about the harm and the benefit, if we don't have enough, the population for phase 3 trial, because you cannot evaluate the efficacy, and if it still deserves to do a human challenge just for the overall good. And so it's an ethic, it's a harm and benefit. And there's one discussion is if we can use some like a weekend virus and to test before we doing the real wild type virus. So there are a lot of discussion about that. So about if how we can do the phase 3 either to the general population or mimic the general population with the human challenge experiment.
So the big problem. Sorry, go ahead, Dr. Cook.
I just want to comment on that, and I thank you for bringing that up, Eddie, and discussing it, Dr. Chen. I just wanted to add, as a physician, scientist, I have problems with the approach of intentionally infecting someone with the virus to test the vaccine, because with this one, because we don't understand it completely yet. And as a cardiovascular specialist, I'm very concerned about the increase in myocardial infarctions, heart attacks, and strokes that we're seeing in patients. And we're learning some things about this virus. It's unusual in that it can invade blood vessels and cause an inflammatory response that may be responsible for these heart attacks and strokes and venous thromboses. It's something that we're studying in my laboratory to understand, what is this virus doing to vessels that it's causing these significant cardiovascular events.
And isn't that what been a problem for some of the younger people who've gotten the virus? Isn't that right?
Yes, that's absolutely right. So there's been some young people with strokes. There's been people with mainly with previous cardiovascular disease, but even some younger patients that have had heart attacks. Usually, though, it's a person with pre-existing cardiovascular disease. That's another interesting thing about this virus is that it's individuals with high blood pressure, diabetes, cardiovascular disease. People that have pre-existing problems with their vessels are more likely to have adverse effects of the COVID-19. They're more likely to have progression. They're more likely to have ARDS, the acute respiratory distress syndrome that causes people to be ventilated. So I think that's an issue.
Well, I guess that gets us then back to the question of treatments. If we could find an effective treatment to treat people with the COVID-19, that would lessen the risk of the bad events and perhaps make a human challenge trial more ethically adoptable. You mentioned the plasma treatment that has been done at Methodist. What other treatments? Are there any other treatments, Dr. Cook, that you think look promising for COVID-19?
Yeah, there are. Remdesivir is an antiviral agent that has been shown to reduce the hospitalization stay, reduce the length of stay in the hospital. And there was a trend toward a 40 to 50% reduction in death. It didn't quite make significance in this study that was done, but there was a trend toward a reduction in mortality. So that looks very promising. There's more trials going on. We've been conducting a remdesivir trial at our hospital. So that's one that looks very promising. The convalescent plasma we mentioned. Also, there's some anti-inflammatory drugs that can help with the cytokine storm that look promising as well. So I think we do have now we have some arrows in our quiver that are giving us, our patients, a better chance in the hospital.
I forgot to mention, you're on the committee at Methodist that's examining the potential treatments and deciding which treatments you might want to try to attempt to use at the hospital. Isn't that right?
I've played a small role in it. It's really the real heroes are the clinicians that are on the front every day dealing with this and making the observations that are helping guide the changes in the therapy. And the amount of information that's coming from all over the world, including our friends in China, has really helped us to guide our therapy, the information that's being shared.
Let me ask Dr. Chen, you've had, China's had the most experience with this disease. Some of the, you've had the experience with SARS and MERS as well. are there any particular promising treatments that you're aware of that are being examined in China that might be adopted more widely?
During the earlier outbreak, I think it has been tried a recover patient, the convalescent sera, and some of the result has been published. Now, in recent weeks, I saw a few monoclonal antibody have been developed, and one of them has been tested in animal models. So antibody specifically targeting COVID-19 may be one of the direction. But for this particular virus, just Dr. Koo just mentioned, is we have to do two things. One is to suppress the viral replication. Another thing is to prevent the cytokine storm. Because once virus is infected, and it turns on the immune system, and it just hijack the immune system and make a cytokine storm. So in our animal model, we also see that is even in the late stage, we don't see many virus replication in the animal line, but animals still go to the very poor condition, and so it's because of the apparent immune response. And we know some of the patients, when they went to the hospital, is already in infection with a few days or into the disease onset. So the immune system already turned on. So we have to do something to inhibit the virus. We also have to do something to intervene with or to try to get the immune system back to balanced level. So that's very challenging. So I think there are many drugs are being tested if we can do that part, and also testing some either repurposed or new drug for suppressing the viral replications. So we have to do two things. The monoclonal antibody may be able to neutralize virus, but may not be able to turn back the immune system. So there's two areas we have to work on. One is to suppress viral replication, and another one is to get the immune system back to the normal level.
Well, that would be great. Let me ask you, Dr. Cook has mentioned a couple of times the collaboration that's going on internationally to attack the virus. Of course, politics have intruded, particular politics between the US and China. Have you, from the trenches, from your perspective, have the sort of the threats of sanctions by the US to China or vice versa, has that inhibited the back and forth that the collaborations that you may have had with American scientists or the sharing of data? For you, Dr. Chen.
Oh, okay. Yeah, from multiple channels, we see there are issues going on between US particular and China. But for scientists, I have not experienced particular difficulties in collaboration. Particularly, we have collaboration mainly is based on individual scientists. So like recently, we still have collaboration with Mount Sinai. So we are testing a few of those drugs. They say they are promising, but they don't have animal model. So we have animal model here. So we are testing there. So we are still working on our collaborations. So for individual projects, I don't see there's any issue. But I don't know, in a higher level, in the government level, there may be some difficulty times. But for individual scientists, we are collaborating with scientists in the US and also scientists in UK. So
well, that's great to hear. So from your perspective, from the trenches, the cooperation internationally is still occurring.
Yeah.
I would agree with Dr. Chen. It's just wonderful for us to behold this kind of collaboration that's global. I will say that I'd just like to take this opportunity to say that in my own neck of the woods, it's really a team effort, and I'm just proud to be representing a team of people, a team of scientists that are really doing the hard work of generating new therapies, new approaches to COVID-19. So I'm really just happy to be part of the team, and I'm happy to be working with scientists around the world on this problem. We have to do it as a team. I will say there is one thing, Eddie, though, that has been a problem, and that has been we have a lot of Chinese scientists working in our institute who are super. I'd like to have more. I'd like to be able to welcome more scientists from around the world. But immigration has been difficult. And part of it obviously is the pandemic, but there's political issues now that are interfering with the free travel of scientists back and forth. And that's only going to have an adverse effect on the whole global community.
Well, I guess we're all going to have to use more Zoom. Well, we were starting to get a number of questions from the audience that's out there, and I thought I would toss a few to you two experts. Donna asks, well, if we get a vaccine, can we produce it in quantities that make a difference? Let me ask you, Dr. Cook, especially about these new RNA and DNA technologies. Will those, if a vaccine appears there, will we be able to produce it in a large enough quantity to get it out there?
Yeah, the answer is yes to that. It's to be sure, there's still scale up going on in the manufacturing process. I know that Moderna and the other companies that are closest to getting their vaccines to market are scaling up, and I think they will be successful. I think what's the issue is more going to be, are they going to be able to address all the regulatory issues? There is one thing though that has been a concern, are we going to have enough medical equipment, syringes and other things like that to vaccinate everybody as quickly as we need to? So that's actually has been a concern. I think that'll be addressed too. By the time we have a vaccine, we'll have those things addressed.
Well, now the RNA vaccine, is it going to be like a shot, like a normal vaccine, or is it another way?
Yeah, actually, there's different approaches. Moderna is putting their RNA into a little lipid nanoparticles, and they're administering it as a shot. That's correct.
Mm-hmm. Now, and the U.S. government, I know, is spending billions of dollars making production facilities already for vaccines that don't necessarily exist. Is that a good investment on the behalf of our government?
I would say this. I would say that the government should make a lot of small bets rather than putting all their money in one pile, which is what they've been doing. I think someone wanted to make a big statement, so they put their money in a big pile. I think it would be smarter to spread the money around. Now, actually, I have to say that is going on as well. The government's making some small bets through the National Institutes of Health. So there's been a lot of money that's gone to the National Institutes of Health and Department of Defense for COVID-19 related projects. So that's a good thing.
Small bets meaning only tens of millions of dollars, right? Still big money. Dr. Chen, what about in China? Will the vaccine technology that you're working on, will it be able to produce vaccine in quantity and quick enough to potentially make a difference?
Yes, for our vaccine is a flu-based. So basically, you can use the flu vaccine manufacture capacity to do that. But we don't know, because even China is a country, the vaccine, the flu shot coverage is not very high, much lower than compared to the US. So if the existing flu vaccine manufacturer may not be able to produce sufficient or enough vaccine, so that's why they have other vaccines. But if they expand the manufacture capacity, it can increase the amount. And for our vaccine, it can be manufactured with the egg, but it can also
fertilized eggs, right?
Yeah, it can also be manufactured from cell cultures. And cell culture is that you can, if you have you have facility for like the cell factory, you can expand and you can not necessary to be very long time to expand the capacity. So we don't say it can be in a few months, but if you expand, I think it can produce in larger amount in relatively reasonable time for general population.
Well, I hate to try to pin you down, but In America, they've been saying things like maybe a vaccine by the fall, or late 2020 or early 2021. How realistic is that timeline, do you believe?
I think we're going to miss the fall.
Okay.
I think we'll have a vaccine by 2021.
Early 2021 or late 2021?
How about mid 2021?
Okay.
I comment on that because we just take the experience of the swine flu. It takes 11 months or 12 months for the first batch of vaccine delivered to human population, and it's a flu vaccine. We have the mature manufacturing capacity. It took about 12 months for the first batch to deliver to general population. And this new virus is a new vaccine, and some of the vaccine are completely new. We don't like a mRNA vaccine. We have not used before, and other vaccines have even the adenovirus, the UK strain, and the strain are now developing in China. We have not used that before, so you need a new manufacture facility. So it may take a little longer than the swine flu. That time we have the 2009 the swine flu outbreak. So it may take a little longer than that swine flu vaccine.
Well, I guess what I'm learning from this conversation is it's unlikely to have one vaccine that is better than every other vaccine in all dimensions. Some vaccines might be better to protect you. Some vaccines might be better, have greater side effects, maybe more efficacious. How do they decide which vaccine to produce? What is the most important quality that they will use to determine, or will they produce multiple vaccines? Will they be giving people multiple vaccines once they're produced?
It's a good question, Eddie. I think that we need multiple shots on goal. Ultimately, the FDA is going to approve a vaccine based on safety and efficacy. So I am quite sure you're right that there'll be vaccines that are more effective than others, and then the marketplace will determine which vaccine becomes predominant. But in the short run, in the near term, I think we'll have multiple shots on goal. By next year, we'll have a few candidates.
Great.
Our supplement on that, yeah, probably because it's different, multiple technologies, and some of the vaccine maybe they can immunize through different routes. So like some of the vaccine maybe just through intramuscular, to induce some of the immunity from antibody and some cell immunity, and some vaccines maybe can be immunized through nasal spray. So like our flu vaccine, it's now tailored for nasal spray. So it may induce mainly the mucosal immunity in the respiratory tract regions. So yeah, different vaccine through different vaccination route maybe can be used for different age group.
So, okay, you mentioned this mucosal immunity. I've heard that phrase used by others. What does that mean, mucosal immunity? Is that, I guess you're in your sinuses or something?
Yeah, basically induce immunity in a respiratory tract, in the nasopharynx, oropharynx area. So it will induce the T cell immunity. It will induce the mucosal antibody like IgA. So it's regional. And because for this particular virus is through the respiratory tract, so we think if we have the regional immunity, it may be the first line to block the infections. So that's a mucosal immunity.
So that's important to keep it from spreading.
Yeah.
Well, one last issue that is raised by Barbara here is that of equity. Once the vaccine is produced, how will we make it available to everyone? Even those countries, the poor countries of Africa, et cetera, who may not have the production facilities. is the strategy in China? Do they have a policy about the equity of the vaccine, Dr. Chen?
Just a few days ago, I heard the speech from the President Xi in the WHO, the General Assembly. He announced the vaccine made in China will be shared by any other country. They don't have vaccine programs. So that's what I heard. And for this particular vaccine, from my personal perspective, I say we have to make the vaccine available to any place they need, because disease doesn't have country border. And if we don't do globally, if we leave some countries still have disease, we are not safe. So we have to do whatever we can to give the vaccine to any places they need that, and only that way we can block, we can control the further dissemination or the recurrence of this virus.
So, for example, if the vaccine that your lab is developing, if it's successful, the Chinese will not put a patent on it or try to limit its use around the world or try to collect royalties if it's used around the world. Have you talked about that yet?
I don't know, because now it's only in the clinical trial stage. It's not commercialized yet.
Right.
Once to that stage, and we don't know, because China now has had very few cases, and it may, if there are some countries have more severe outbreak, and if I can decide, it may deliver those vaccines to those places to contain the outbreak first, and then to other places to see which place are more need vaccine for control the outbreak.
Okay. Dr. Cook, we have, of course, a very difficult system here in America about the distribution of healthcare. What comment would you make about how the vaccine is successful? I mean, Inovio and Moderna and all these companies we mentioned are for-profit, and lots of investors are invested in them and hopeful that they will be able to make a lot of money on these vaccines. Does that mean that they're going to have to restrict the supply?
So I agree with Dr. Chen that it's the right thing to do, and it's just good international policy to make the vaccine available for everybody. You have to figure out a way to do that, and still for the companies that are for-profit companies, you still have to make money, because without a margin, there's no mission. So yes, the companies you mentioned, Inovio and Moderna, they're hoping to make money on their product, and that's our system, and it's a system that seems to work. It does spawn innovation. So we have to find a balance between supporting the mission with profit, yet make available to everyone the vaccine. And I think philanthropy like the Gates Foundation and government assistance, those programs have the World Health Organization. Those programs have made vaccines for other diseases available worldwide.
Well, on that hopeful note, I think we could probably talk all night about the different candidates for vaccines, but I think it's probably a good. We're at our the end of our time here, and that's a hopeful note to end on. Thanks to everyone who've joined us from Texas, Hong Kong, around the world, and those who may sign on to see the recorded interview in the future. Before we leave, I'd just like to mention that both Hong Kong and the Texas Center will be continuing on our COVID-19 series next week, at least in Texas. It'll be at 7:30 p.m. Central on June 4th. The topic next week is the second wave of COVID-19 cases inevitable? We will feature Dr. Ali Khan, author of The Next Pandemic and Dean of the College of Public Health at the University of Nebraska Medical Center. He also served as the former Assistant Surgeon General with the U.S. Public Health Service. Dr. Mark Boom, President and CEO of Houston Methodist, will join him in this conversation. The Asia Society Hong Kong Center will also be hosting 2 programs on COVID-19 coming up. Their next episode on June 2nd is with Dr. Jeffrey Burns, Chief of Critical Care at Boston Children's Hospital, on the topic of children and COVID-19. That's obviously a very hot topic right now. Dr. Burns is spearheading international efforts to look more closely at COVID-19's impact on children and the rare pediatric inflammatory multisystem syndrome that has unfortunately come up. On June 9th, they will host Professor Peter Horby of Oxford University. He's leading the largest COVID-19 recovery trial in the world, treating over 5,000 patients in connection with 165 UK National Health Service hospitals. So both of those sound to be very interesting. Until then, thanks to Dr. Chen and Dr. Cook for joining us. And thanks to all of you out there for listening in. Until we see you again, thanks so much. Bye-bye.
Thank you.
Bye-bye.